PhyBits are minimally animated supplementary videos that for one reason or another couldn't make it into the main video. This one's another follow-up to the Prion video I made a while back, and I'm finally addressing some comments that have begun to build over the course of these last few months. In this video, I made the claim that it was unusual that the Prion gene has been so well conserved despite not having any obvious important use. Progression in prion research has now allowed us some insight as to what the true cellular function of the prion protein is. It just wouldn't make sense for evolution to have conserved this gene across so many walks of life in spite of its potential for biophysical corruption. If it were truly useless and truly just harmful, evolution would have likely jettisoned this gene from the genome long ago. If you delete the prion gene, then mice are largely normal. Nature doesn't like to maintain parts that it has no use for, and the fact that the prion gene seems to be pretty well conserved across mammals indicates that it has to have some kind of useful function, just one that can't be detected easily. Many commenters have pointed out that selection works primarily on traits that enable successful reproduction. This concept is the concept of fitness. How much does a gene contribute to the successful passaging of your genetic material? If the prion gene doesn't do anything, then it makes sense that evolution would not select against for having the prion gene. And that's kind of true to a certain degree, but what these commenters are missing is the fact that entropy exists. Order must be actively maintained. Everybody over 30 understands this in a visceral way. Even if an allele is not actively selected for or against, changes in the gene are still expected due to mutations. Remember that every time DNA is replicated, there is a chance that something goes wrong and a mutation is introduced. If a gene is truly useless, doing nothing at all, then you'd expect this chaotic force of entropy to introduce variations into the gene penalty-free. There is one exception though, and that's if mutations in a gene turn a useless protein into a protein that does active harm. And in the case of the prion gene, this can be a contributing factor. It is known that genetic mutations in certain regions of the protein can increase the chance of misfolding, and if you've watched that prion video, you know what the penalty can be. This explains why the C-terminal end of the prion protein is so highly conserved. Break this part, and well, you're in for a bad time. But this is just a story that makes intuitive sense. Break up the C-terminal in unexpected ways and you get prion disease. Makes sense why nature wouldn't want that, but again, the commenters have a point. If it doesn't affect your fitness at reproductive age, then this shouldn't be selected for. Indeed, a more recent analysis zooming out on prion variants across species reveals that mutations in this prion folding region don't really influence how the prion protein evolved. So, to sum, the prion protein is well conserved across mammals, and that's kind of weird. If it were truly useless, you'd expect the forces of entropy to cause greater variety in this protein across species than what we actually see. This lack of genetic diversity cannot be attributed to mutations in the prion gene being toxic, and it is unlikely that a purifying force like genetic drift would affect all mammals to this degree. So with alternate hypotheses more or less ruled out, we have one more staring us right in the face, that the prion gene is doing something useful enough that evolution puts in the effort to limit the amount of variation that can exist within this gene. This is supported by the finding that the regulatory elements flanking the prion gene are also pretty well conserved. If conservation was just about limiting prion toxicity, you wouldn't expect the stuff that determines whether a healthy prion protein gets made to also be functional. In fact, if the prion gene were truly useless, at some point these genetic regulatory sequences would decay as well. I mentioned in my video what the prion protein could be doing, stuff like regulating myelination of neurons. But I think for many of you this wasn't sufficient, so let me try to do some hypothesizing. If the prion protein is involved in some complex reaction to an adverse life event, mutating it in a lab strain of mouse wouldn't show significant differences in fitness. But given we know the prion protein can regulate myelination, what if it did so only in the context of brain injury? This is a case that can affect reproductive fitness if it happens early enough and would be missed in a lab setting unless brain injury was specifically studied in mice. I don't know if this is true or not, but it is one hypothesis that could explain why the prion protein is important, but easy to miss in a research environment. Hope this helped.